Hello, everyone, and welcome to today's webinar, Streamlining RNA Analysis, Eliminate Traditional RNA Purification with Invitrogen Cells-to-Ct technology. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, today's presentation is interactive, and we encourage you to participate by submitting as many questions as you have at any time during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box, and we will assist you directly. I would now like to welcome our speaker, Laura Meyer, Senior Manager, R&D Sample Preparation, Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Presenter tab at the left of your screen. Laura, welcome. It's so wonderful to have you. You may now begin your presentation.
Thank you, Christy. And thank you, everyone, for joining me today. And I'm excited to talk to you about how you can streamline your RNA analysis using our cells-to-Ct technology. So first, a quick overview of our agenda for today's webinar. We'll go over what gene expression is and RNA and why it's important, and then how the cells-to-Ct technology works. There are multiple options that you can choose from the cells-to-Ct technology, so we'll go over different workflows that you can choose from. And we also have some specialty kits that I'll cover that may be able to help advance your research. And then lastly, as Christy mentioned, we'll have some time for Q&A. And so with that, we'll get started with the webinar. So first off, what is RNA expression? Generally, RNA is used to look at a snapshot in time of what's going on within a sample. It can be used to look at the gene expression as to what's going on, and then it also can be analyzed over time so that you can see what is happening within a time course. Perhaps if you're doing some kind of treatment, you can see what happens over time. And it's often used as a research tool to look for biomarkers for specific diseases, or perhaps what's happening with a small molecule that is affecting your sample. There are some challenges of working with RNA. If you worked with RNA, you're probably well aware that it is temporal due to exogenous and endogenous RNases that are around. It will quickly degrade your RNA, and so extra precautions need to be taken to make sure that your RNA will stay intact and that you can trust your data in the gene expression analysis that you see. Some downstream applications that are often used for RNA are real-time PCR. This is very popular. It's really easy to use, and it allows you to see both quantitative expression of your RNA, or it can be used for a presence-absence detection of a specific gene that you're looking for. It can be used in both TaqMan and Cyber options, depending on what your lab would prefer. Another popular option is next-generation sequencing. Thermo Fisher offers the ION brand for looking at next-generation sequencing, where you could do whole transcriptome analysis of your RNA. But the market leader is Illumina, and these are both beneficial to look at high sensitivity and to look for new genes. So if you're doing more discovery work, you would want to use something like next-generation sequencing to look for genes, whereas if you know what genes you're looking for, then you could use real-time PCR. Another option that's not very popular is to do gel analysis, where you'd be able to run your RNA on a gel and look at the integrity. But a more popular option would be to use something like the Agilent bioanalyzer so that you could see the integrity of your RNA and make sure that the quality is good. These are some of the applications and kits and reagents that I'll be discussing today. Don't need to take note of these now, but just wanted to quickly highlight what ones we will be discussing. And so with that, we will go into what the cells-to-Ct technology is and how it can advance your RNA research. So this is a high-level overview of the cells-to-Ct workflow. As I mentioned, it's a way that you can streamline your RNA analysis because it eliminates the need to purify your RNA. So this is intended to work with the mammalian cells, and so you would start with your cells in a culture plate. Here is an example of a 96-well plate, culture plate, in the first image on the left of the workflow diagram. You'd remove the media from those cells because some of the medias may have inhibitors into your RT-PCR, so you would need to remove the media, wash with PBS to get rid of any residual media, and then remove that wash, and then add a lysis buffer. The lysis buffer can be pre-mixed with a genomic DNase to remove any genomic DNA in your sample at the same time as your lysate is preparing the RNA. That would incubate at room temperature for 5 minutes, and you would need to pipette mix or either use an orbital shaker, which is a little bit easier and provides a really great mixing method, and allow that to sit for 5 minutes. We have 2 different versions of cells-to-Ct. Some of the older kits would require a stop solution, but our newest and cells-to-Ct kit, which I'll go into more detail later, does not require this additional step. But you would add the stop solution, mix that, and let it sit for 2 minutes to inactivate the lysis reagents. And then from there, you could choose whether you wanted to do 1-step or 2-step. And then we also have cybergreen or TACMan technologies that you could use for your detection method. So the real benefit of this is that it's very easy to use, and it eliminates the tedious work of extracting your RNA and purifying your RNA. It works for most mammalian cell types, and I'll go into more detail later as to which ones we've tested. And it's a really simple technique for being able to get to your RNA analysis from cells. Some more of those benefits is that it was completely optimized together. So the reagents, the lysis reagents, the RT, and the qPCR master mix were all optimized to work together to give you the best and most sensitive detection for your RNA from your samples. And the other thing is that it's that I mentioned previously is it's very fast and it's very easy. The formulations for most of the reagents included in the kit are in a master mix format. So you don't have to worry about mixing individual components together to create your RT master mix, for example. And so this helps minimize errors from having to use multiple tubes and multi-step protocols. And lastly, the sample preparation is very easy and very fast. It only takes about 10 minutes or less than 10 minutes, depending on the number of samples. And it's easily automatable. As I mentioned, everything's at room temperature, so that makes it very easy to add onto liquid handlers, and you don't have to transfer into different like incubators for heating or anything like that. The genomic DNA is also removed simultaneously with that DNase that you add into the lysis buffer. So your RNA is prepared as while as the gDNA is being removed. And another benefit is that can be done directly in your culture plate, so you don't have to worry about accidentally mixing up your samples by transferring into another vessel. And again, there's no centrifugation steps, and so that makes it really easy. It's all done at room temperature. There's no organic waste that you need to worry about, so no hazardous waste to deal with. And then for spin columns, there's the potential that they could clog, and so you don't have to worry about this here because there's no spin columns involved. Another major advantage of the cells-to-Ct technology is that it's much more sustainable in the fact that it uses a lot less plastics, and it also does not generate the hazardous waste, as I mentioned. And this image is an example of the amount of waste that would be generated from 12 cells-to-Ct express lysis reactions on the left side, which is a very small amount of plastic. And then on the right side is the amount of plastic as well as the liquid hazardous waste that would be generated from 12 spin column reactions. And this is a savings of about 95% plastics from the spin column as compared to the cells-to-Ct express kit. So you're not only speeding up your research, but you're also spending less money on the plastics that would need to be used. As I briefly mentioned earlier, there are different workflow options that we offer with the cells-to-Ct technology, so I'll go over a few of those kits now. So these are 3 of our main options that we offer. We do have a few others, but these are the 3 that I would recommend to start with if you're just getting into the cells-to-Ct technology and are interested in using it. The first one is our cells-to-Ct Express Kit. This is the newest product. It's been on the market for about a year, so it's our newest one. It's our latest and greatest, and the fact that it does not require that stop solution, as I mentioned earlier, so it's one less pipetting step that's required. And it also uses the superscript 4 Bilo RT for ultimate sensitivity for your RNA transcripts. And it includes also the fast advanced master mix for your qPCR detection. And this is the same master mix that you could get as a standalone item. So if you needed more master mix to look at potentially more genes, then you would be able to buy more of that, and it would be the exact same master mix. The other kit I'd like to highlight is the TaqMan Fast Advanced Cells-to-Ct kit. This one has been around for several years, but it's a tried and true method. And one thing that we do know is that it works for spheroids. We have not had the opportunity to test the Express kit with spheroids, but we do know that the Fast Advanced kit does work with spheroids, and so you could potentially use it for your 3D cultures. It has a slightly older RT master mix, so it doesn't have the superscript 4 Vilo, but it also includes the same Fast Advanced master mix that's included in the Express kit. We also offer a cyber option for the Fast Advanced, so if you're interested in cyber, this could be an option for you. And then lastly is the Cells-to-Ct one-step kit. This one is going to be your fastest workflow because you can skip the RT step. It's included in your qPCR reaction. It has the same lysis technology as the Fast Advanced kit, but as I mentioned, includes that one-step master mix. And so if you're interested in like really speeding up your research, the one step is probably the best method for you. So the Cells-to-Ct Express kit has been tested with multiple cell lines, and we have an application note and the link listed below that you would be able to see more details. But we've tested with these particular cell lines here listed in the table. We tried to use a wide variety of cells from different sources to be able to show that it's compatible with a lot of different mammalian cells. This doesn't mean that it's not compatible with other cells on this list. It just means we haven't tested them yet. So if you are interested in trying it out with your specific cell line, I think that you would probably likely to have success. You would just need to test it and see, because we have not had the opportunity to test any others besides what's listed here for the Express kit. For the Fast Advanced and one-step kits, those have been out on the market for a little bit longer, and so we have had the opportunity to test more cell lines with these. And listed here are the cell lines that we've tested with these kits. So it's a few more than the Express, also from a wide variety of different sources. And again, this doesn't mean that it wouldn't work on your specific cell line. We would just need to test that in your lab with your conditions and see if it would work. This is an example of the experiment that we did to confirm whether the cell line was compatible or not. And the details of how you can process this experiment is outlined in the Cells-to-Ct Express user guide. But briefly, using Raji cells, we serially diluted the cells so that we had 10 to the 5th all the way down to 10 cells per 5 microliters of PBS, and then added the Express lysis reagents, including the DNase, to remove the genomic DNA at the same time. We also took separate aliquots of those same cell dilutions and purified the RNA using a spin column kit. And because we eluted the RNA in the same volume as the final volume of the lysate, we were able to add equal volume into the RT, into the qPCR, and then directly compare the CT values. And our criteria for showing that it was compatible with the Cells-to-Ct Express was that there would be no more than 1 CT greater than the purified RNA, and that the slope would be minus 3.3 within plus or minus 0.3. And the reason why the slope matters is minus 3.3 is considered a perfect slope for 10-fold serial dilutions in qPCR. And so as you can see here on the graph on the right of the screen is that we see equivalent or better performance with the Cells-to-Ct Express kit as compared to the purified RNAs via spin column. We get lower CTs with all of the cell inputs. And then as we decrease the cell input, so when you get down to like 10 to the 3rd or 10 to the 2nd or even 10 cells, you see a greater difference between the Cells-to-Ct Express versus the spin column. And this is because whenever you're purifying RNA, you don't lose, or it's not 100% efficient at each step. And so you could potentially not be binding all of your RNA, losing some in the wash solutions, or not eluting all of your RNA at the end. And the less starting sample that you have, the greater that impact has. And so as we decrease the amount of cellular input, the greater that impact of not 100% efficiency has on your detection. And so that's another great benefit of Cells-to-Ct is that if you're using a really small number of cells, you don't lose any because you can process your samples and lyse them directly in your culture vessel, and you don't lose any by transferring your samples from vessel to vessel. And then this is a little bit more detail about the slope that I mentioned. So here we did the cell line compatibility experiment, this time looking at skNAS cells. And again, we did the 10-fold serial dilutions in PBS and then either processed using the Cells-to-Ct Express kit or using a purified RNA spin column kit. Here, the red line indicates the CT values from the TaqMan cells-to-Ct express kit, and the blue is from a spin column. And again, we see that separation as we decrease the cellular input. And again, it's because the RNA is not 100% efficient at every step. And so you see a wider variance at that lower input than you do at the higher input. And again, the slope is, as I mentioned, a minus 3.3 slope is considered 100% efficiency because that would be ideal for 1 to 10 dilutions. And so the slope is more negative with the spin column kit because we're getting higher than expected CT values because of the inefficiency of the RNA purification method. And I briefly mentioned that it's very easy to automate, and it's because it has such a simple workflow. You don't need to do any centrifugations. There's no heating steps and no incubation steps at different temperatures. So it makes it really easy to automate. And so we did an experiment here where we plated 5,000 HeLa cells per well in a 96-well plate and duplicate 96-well plate, and then process the 96-well plates either manually or using the TCAN Fluent 780. And then because we eluted or because they had equal volume for the lysates, we were able to add equal volume to the RT and qPCR reactions so that we could directly compare the CT values. And so on the graphs on the upper panel is the amplification plots shown for the manual versus using the TCAN. And so you can see that they're both really tight, and we can see a nice separation between the two different genes. We looked at CDK4 and beta-actin as the two genes of interest for these particular samples. And so the amplification plots look really nice for both manual and the TCAN. And then those CTs were average for both the manual and the TCAN automated version. And those are graphed in the graph below. The left columns are your beta-actin, and then the right columns are CDK4. And you can see between the manual method in the red bars and the automated method in the gray bars that we get equivalent performance between the two for both genes. And so you're able to easily automate. And so it would be a great tool if you're doing high throughput and have multiple 96-well plates that you want to process. And this is a use case if you wanted to use cells-to-Ct for some type of screening. Here we're looking at RNAi screening. And so HeLa cells were plated in our 96-well plate and reverse transfected with 20 different siRNAs and using a positive control of GAPDH for our positive control siRNA. They were grown for 48 hours, and the knockdown was assessed by doing reverse transcription and qRT-PCR. And then the relative gene expression was calculated, and that's what's indicated here on this graph. The orange bars are the cells-to-Ct, and then the green bars are a duplicate plate that was done using purified RNA. In this particular example, we used a magnetic bead-based purification for the RNA. And again, we see equivalent performance across all 20 siRNAs indicated on the x-axis, and our positive control GAPDH is the last set of bars. So you would really be able to accelerate your gene expression or your screenings for our siRNA or perhaps even small molecule screenings on cells if you use the cells-to-Ct technology, because with the magnetic bead-based kit, which is generally faster than a spin column method for multiple samples, here it took about an hour to process the samples, whereas with cells-to-Ct, it only took about 10 minutes to process the samples. And so it was an 80% time savings as compared to purified RNA. So you can really advance and speed up your research using this tool. We also wanted to confirm that the cells-to-Ct technology would work with a wide variety of TaqMan assays, and that you would get similar performance across a wide variety of expression levels, as well as like GC content and difficulty to amplify assays. And so with that, we tested by processing 10,000 HeLa cells with purification of RNA using a spin column, or we lysed the cells using the cells-to-Ct express kit. We put equal volume into the RT, and the qPCR was done using the TaqMan array cards. If you're not familiar with those are microfluidic cards that you're able to process many wells or many different assays with one sample. And so we're able to process 96 different TaqMan assays with the one card, and to compare to cells-to-Ct express and the purified RNA. And those CT values were plotted here with the y-axis indicating the purified RNA and the x, the cells to CT expressed. And then we did a correlation, a fit line to see how well they correlated. And we have an R-squared of 0.96. And so we see really nice equivalency between purified RNA and the cells to CT lysates here with 96 different TACMAN assays. The next thing we wanted to do was to confirm that with cells to CT, you're able to detect low copy genes in a background of high expressing genes. And so the way that we tested this was we spiked in an increasing number of NIH3T3 or mouse cells into a constant number of HeLa or human cells. And so we had 10,000 HeLa cells and spiked in 1,000 all the way down to one NIH3T3 cell, and then tested for a mouse-specific B2M gene, or we looked at the human-specific TKT gene for both purification for both purified RNA using a spin column kit or using the cells to CT technology. And so the data shows that with the orange and green lines that are going across, that is the human-specific TKT for both purified RNA and cells to CT lysates, that we get equivalent CTs across all the cellular input for both methods. So the high expressing gene is detected equivalently across. And then as we spike in more and more NIH3T3 mouse cells, you can see the CTs decrease with the more cellular input, and we're able to detect down to 10 cells in a linear fashion. But then even down to one cell, we're able to detect it, but it's kind of at the limit of detections there. It's almost at background level. But this is a really nice experiment that we did to show that low copy genes are able to be detected in a high copy background in equivalent performance to purified RNA. So next, I'd like to go into some specialty kits. These may be beneficial for your lab or maybe someone that you know that's working with some of these specialty sample types. So some of the kits that I would like to talk about today are using stabilized blood to CT. This particular kit was optimized to be able to look at RNA expression from stabilized blood tubes, such as the PAXgene blood tubes or the Tempus blood tubes. These tubes are collection tubes, so the phlebotomist would collect the blood into those tubes, and then the stabilization reagent stabilizes the RNA in the blood because there are lots of RNases in blood, and so these tubes will help protect your RNA for processing. You're able to process 96 samples using the stabilized blood to CT kit in about an hour. It uses 500 microliters of your sample size. And so you don't have to use your entire stabilized RNA or stabilized blood to look at a quick look at your RNA. You'd have the majority of your sample left over if you wanted to process for other downstream applications. And because stabilized blood is a lot more challenging sample to work with, this does require some centrifugation and wash steps to help remove some of those inhibitors and proteins that come along with the blood sample. The next kit that we'll talk a little bit more about is the single cell to CT kit, which allows you to look at single cell gene expression by a qPCR. This uses the superscript Vilo RT master mix, and so it allows for ultimate sensitivity for your low input samples. And it also includes a preamp master mix that will help you amplify your cDNA, but prior to going into qPCR, so that you don't have to worry about running out of cDNA from your precious sample. And then lastly, we'll talk about the cells to CT control kit. This includes an exogenous xeno-RNA that has no known homology to any known gene. And so you're able to use that by spiking into your lysates to look for inhibition or incomplete lysis. And the kit also includes not only the RNA, but it includes the TACMAN assay for looking at that specific xeno-RNA. And it also includes a housekeeping gene assay, beta-actin. So this is a workflow of the stabilized blood to CT kit so that you can get a little bit more detail as to how it works. So first you would have the blood collected in your stabilization tube, whether it be Tempus or PAXgene tube, and that allows the RNA to be protected from the RNases. You take an aliquot of that, and then with this particular kit, there are two options that you can choose from. If you're doing a low throughput and you only have a few samples, you could use tubes where you could put the sample into the tube. Or if you're doing a high throughput and you have lots of blood samples that you want to process, you could also use a 96-well plate method. And so you would aliquot 500 microliters of that stabilized blood into either the tube or the plate format, depending on what fits your needs. Pellet those and then wash the pellet a couple of times to get rid of any inhibitors that have come along with the blood. And then you resuspend in the digestion solution and incubate that for 8 minutes at room temperature. Once the incubation time is up, then you would add the stop solution, allow that to sit for 2 minutes to fully inactivate your lysis reagents, and then you're ready to go into RT-PCR. And so this can really speed up your stabilized blood RNA expression analysis by it only taking about 60 minutes to process either 12 or 96 samples if you're using the blood-to-CT kit. However, if you're doing a purification method, it would take about 2 hours to process 12 samples or 4 hours to process 96 samples. So it's a huge time savings for processing stabilized blood. This is some example this is some example data from gene expression of using the stabilized blood-to-CT kit. In this particular experiment, we had a 10 donor pool of Tempus stabilized blood, and we took 500 microliter aliquots in duplicate and prepared the duplicates either using the stabilized blood-to-CT kit or the Tempus Spin RNA isolation kit, and then compared the purified RNA to the lysates for both microRNAs or mRNAs. And so the graph in the middle here on the left side shows the mRNA expression profile of lysates as compared to purified RNA. And one other thing that we tested here was comparing the tube purification or the tube preparation method and the 96-well plate preparation. And so the tube method is in the blue squares, which are kind of behind the green triangles, which is the plate samples in that first graph. And so because the green triangles are right on top of the blue squares, that means that the 96-well plate version and the 1.5 milliliter tube version for processing your stabilized blood samples work equivalently well. And so you could choose either method and be able to get the same answer. And then by looking at the line between the x-axis and the y-axis, we show that the purified RNA and the lysates perform equivalently well. So you would be able to process the lysates and get the same sample or the same answer that you would expect to get from the purified RNA. And the R squared is indicated in the blue and green for both the plate and tube versions in that graph. So they both provide an R squared of 0.97 as compared to the purified RNA. And then the far right graph is looking at microRNAs. For this one, we looked at 30 individual microRNAs and then compared the lysate average to the purified RNA CT average. And here again, we get really great performance in comparison to the purified RNA with an R squared of 0.95. And so these results show that you would be able to use the stabilized blood-to-CT kit and expect to get equivalent performance as to purified RNA in a much shorter time frame and get to your answer much quicker. The next specialty kit that I'd like to discuss is our single cell to CT kit. And this allows you to look at really small sample sizes, especially for single cells. Single cells are really exciting because you can see what's going on at the individual cell level. We know that that a mass of cells that are pooled together, they don't all have the exact same expression level. And so whenever you're looking at, say, 10,000 HeLa cells, you're looking at the average expression level. So if you look at the individuals, you can really see what's going on with those individual cells. So the method for this is fairly similar. You would have your cell lysis. And for this particular kit, the way that we have generally done it is we fax sorted individual cells directly into the lysis buffer. And here you're only using a small volume of only 10 microliters of lysis buffer so that you can really concentrate your sample into your that final lysate. So we generally fax sort the single cells into the lysis buffer, and then you would add your RT reagents on top of that using the superscript Vilo RT master mix. And then we also include in this kit the pre-amplification master mix, and this allows you to pre-amplify the cDNA for the genes of interest that you have so that you have plenty of cDNA to do your real-time analysis. The major benefit of this kit is that all of the steps are additive, so you don't have to worry about losing your precious sample by transferring from one tube to another tube. So your entire lysate is, the RT is added into your entire lysate, so all of that gets reverse transcribed, and then your preamp is added on top of that, so your entire sample goes into the preamp reactions. We've also shown that it's compatible with 384 well plates and not just 96 well plates. And there are microRNA as well as DNA protocols for single cell kits. This is an example of some data that really highlights the intriguing nature of single cells. As I mentioned, they are each individual cells and they respond differently and they have their own expression profile. And so here we FACS sorted either 100 HeLa cells into lysis buffer, or we FACS sorted individual cells, 84 individual cells into lysis buffer. And then that 100 cell lysis, we did a 1 to 100 dilution, and those are our one single cell equivalents that we tested. And then here we did the preamplification on multiple genes, but did qPCR just on our beta-actin housekeeping gene to see what the biological variability would be. So with the 100 cells, we see an average CT value of about 13.6, and it's really nice and tight, and it shows very little variability, which is what we would normally see with a pool of cells. And then when we dilute that 1 to 100, we see a 6.6 CT difference, which is what is expected for a 100-fold dilution, is a little bit over a 6 CT difference. And we see that with the single cell equivalents. And we also see that it's very little standard deviation there and that we get a similar result between our replicates as expected. But then when you look at the individual 84 single cells and the beta-actin expression, you can see a huge variance in the expression level of beta-actin from those individual cells, ranging from a CT of about 19 all the way up to about 27. So we have like an 8 CT difference there, which is a huge variance in the individual single cells. And this is looking at beta-actin, which is typically considered a housekeeping gene. So even within a gene that you would expect pretty similar expression levels, you see a wide variance at the single cell level. And so this really highlights that individual cell biological variability. And then the last specialty kit that I'd like to discuss is our cells-to-Ct control kit. As I mentioned, this includes a xeno-RNA, which is it's an RNA that has no known homology to any existing gene. And so you can use it to spike into your samples to look for inhibition or incomplete lysis. The xeno-RNA should have CTs that remain constant for samples that do not have inhibition. And I'll show you some data as to what that would look like in just a minute. The kit also includes not only the xeno-RNA itself, but the xeno-RNA assay, so you'd be able to interrogate for that, as well as beta-actin as a housekeeping gene for normalization. And so this is an example of the experimental data that you would see whenever you were using the xeno-RNA as your control RNA. In this particular experiment, we serially diluted HeLa cells from 10 to the 5th down to 106 cells per 5 microliters of 1x PBS. And then we lysed with the fast advanced cells-to-Ct lysis buffer, and then performed RT-qPCR for beta-actin, which is the housekeeping gene that's included in the kit, the xeno assay, which is included in the kit, and then our gene of interest here, which was VEGF. And so the xeno assays are indicated with the blue squares, and you can see that those are equivalent no matter the cellular input, showing that there's no inhibition with this particular cells, with the HeLa cells, no matter the cell input, even up to 100,000 cells, there's no inhibition. And we also see a really nice linear line for the dilution of cells with the beta-actin and the VEGF. Which is really nice to see because the beta-actin is a really highly expressed gene, as well as the VEGF is a much lower expressed gene, but we see that there's no inhibition with any of those because we get nice linear lines and the xeno assay stays constant across all cellular input. But as I mentioned, you're able to detect whether with this xeno-RNA, you would be able to detect if there's inhibition or incomplete lysis. And so this is how you would be able to distinguish whether you have inhibition or incomplete lysis if you're using the xeno-RNA. The graph on the left is an example of what you would see if you have incomplete lysis. And the way you would be able to determine that it's incomplete lysis is with the xeno-RNA, you have consistent CT values across. There's not showing any increase in CT value as you increase the number of cells. However, you do see that the line for beta-actin does not remain linear at the cellular input, at the high cellular input. So the 10 to the fourth and 10 to the fifth are almost the same CT values. And so you only get a linear response down to 10 to the fourth cells. So for this particular example, what you would want to do is add no more than 10 to the fourth cells per lysate, because you're not going to be able to completely lyse if you add more than that. And then on the right side, this is an example of what you would see if you have RT or qPCR inhibition. And the way you would be able to determine that is by looking at the xeno-RNA. So again, we had a serially diluted series of HeLa cells from 10 to the fifth all the way down to 10 cells. I'm sorry, this wasn't HeLa cells. This was a different cell. But what you see is that with the xeno-RNA, you see that it has the same CT value for the lower input, but then at 10 to the fourth and 10 to the fifth, you see elevated CT values as compared to the lower input. And then also when looking at the beta-actin, you see a linear response all the way to 10 to the third cells, but at 10 to the fourth and 10 to the fifth, that line starts to plateau a little bit. And so because the xeno-RNA CT values are not the same all the way across and the beta-actin is also not linear, this indicates that there is qRT-PCR inhibition. And so the solution for this would be to add less lysate into your RT and or your qPCR reaction. So in summary, the cells-to-Ct kits are really beneficial if you're doing RNA expression analysis from mammalian cells. The cells-to-Ct kit would be able to speed up your research because it's much more streamlined than doing a purification method. It's automatable, it's user-friendly, and it also generates a lot less waste, plastic waste as well as the hazardous waste, hazardous liquid waste that you would generate from a spin column purification method. There are multiple options that you can choose from, either a one-step or a two-step, depending on which one you would prefer. And there's also versions of TaqMan and CyberGreen for your detection qPCR. And lastly, there are multiple specialty kits that are offered that may help you with a specific type of research that you're doing. I highlighted here ones for stabilized blood and for single cell, and then also the tool that's really beneficial for looking for inhibition or incomplete lysis within your samples. And so with that, I'll close and take any questions that you may have. And thank you for your time.
Laura, thank you for that excellent presentation. So yes, it is now time for our live Q&A portion of our webinar. To our audience, if you haven't already done so, submit those questions now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So Laura, we've already got so many great questions coming in. So I'm just going to start right here at the top. Are these kits compatible with RNA stabilizers like RNA Later, RNA Shield?
We have some preliminary data looking at RNA Later, where you would still need to wash, and you would need to pellet the cells, wash and remove that stabilization reagent, and then also do the PBS wash. And we showed that that works pretty well. I've never tested with the RNA Shield, so I'm not sure about that one. But the RNA Later, we have some preliminary data that it would work.
Excellent. Are cells-to-Ct kits compatible with both adherent and suspension cells?
Yes, they are. The adherent cells are a lot easier to work with because you're able to vacuum or pipette off the media, and also for the PBS wash that's indicated, so they're a lot easier to work with. But you can also use them for suspension cells. But suspension cells in general are just more challenging to be able to pellet and wash because they don't adhere. And so you would be able to use them with suspension cells. You would just have to centrifuge those down to be able to kind of pellet them in the edges of the wells. remove the media, and then again wash with PBS, have to centrifuge again to kind of pellet them in the edges of the wells and remove that. And then from there, the workflow is the same. Just add the lysis reagent and then continue on with your RT-PCR.
Very good, thank you. So our next question is 2 parts. Are you able to use different RT and qPCR master mixes with the lysis solution? Or does the solution only work using what is provided with the kit?
We recommend only using what's provided in the kit because it was optimized to work together. We did a lot of work to make sure that we found the RT and the master mix that was going to give the ultimate sensitivity. And so I wouldn't recommend that you swap out. You could potentially try other RTs and master mixes if you wanted, but we don't have the data to show how well it would work. So I would definitely recommend sticking with the one that's included in the kit, and it should give you the best performance.
Now, Laura, how can this project be utilized in oncology?
That is a great question. I do know that there have been some folks looking at, like I mentioned, for the Fast Advanced kit, looking at spheroids. And so that could be an option that you would use for oncology if you're looking potentially at tumoroids. We haven't done a ton of work with those particular sample types, but that could be an option where you would use here. You could potentially generate your spheroids or tumoroids with your oncology samples or cell lines that you're using, and then be able to potentially treat with different drugs and look at the gene expression analysis there. The other thing is that you could look for specific cell lines of the cancer that you're researching, and then potentially look to see like what potential drugs could affect the gene expression, or if there's anything else that may be interesting with gene expression from the particular cell line that you would be interested in. You could use it for that. So those are the applications I can think of off the top of my head. I think probably a screening type of workflow would be the best option to use for oncology.
Thank you, Laura. Next question is also 2 parts. How does cell density affect the efficiency and accuracy of the assay? And is there a minimum and maximum cell input required for optimal results?
This is another good question because it very much depends on the cell that you're using. As I mentioned in the presentation, we tested with a lot of different cells from a wide variety of sources. And so because we haven't tested with everything, you would probably need to know for your specific cell what is the maximum amount that would go in. In the user guides, we have what we call a pilot experiment, and that's what we did to validate these specific cell lines that they work with the kit. And so in general, what you would do is perform a serial dilution of the cells. We recommend no more than 10 to the 5th. In general, that tends to be the cutoff. So 10 to the 5th would be the maximum input into each lysis reaction, all the way down to 10 cells. Once you get less than 10 cells, the sensitivity can be a little not great. And so I wouldn't recommend more than 10 cells. So that's kind of the broad dynamic range that we would recommend for the kit. And then for your specific cell, you would need to do that pilot experiment where you serially dilute from within that range to determine if you can detect the gene that you're looking for and your housekeeping gene, and make sure that you're able to identify what it is you're looking for within that range that would work. And then also you could use the control kit to make sure that you're getting that cell lysis efficiency, that it's being 100% lysed, and that you're not seeing any inhibition. That xeno-RNA would be able to indicate that as well, and also help you really determine the maximum input and the minimum input. Minimum is usually by looking if your gene of interest is detected or not. The maximum input would generally be because of inhibition or incomplete lysis. And so the combination of that pilot experiment and using the control kit would really help you answer that question the best. So the answer is it depends.
Very good. Thank you, Laura. And it looks like we have time for just a few more questions here. What is the typical or average turnaround time for completing a screening assay when using cells-to-Ct?
This one, again, it's kind of it depends answer, because it depends on the number of plates that you're trying to process. What we generally do recommend is with cells-to-Ct, the timing is pretty important. You don't want to have the lysate sitting around too long. And so you would want to process one plate, get the cell lysate complete, and then go into your RT-PCR. And then you want that to, so the timing in general that we give is that the lysis is complete within about 5 minutes. And then you want to process the samples and not let them sit around at room temperature for more than about 20 to 30 minutes, depending on which kit you're using. And so depending on if you're using liquid handlers or if you're processing them by hand, that can kind of gauge how many samples you would be able to process within those time constraints. So in general, I would say the average turnaround time for like 196 well plate is it would take only the 5 minutes of lysis to do. And then you would be able to do your RT. Our fastest kit is the express kit. And so that RT is about 25 minutes. And then the qPCR portion of that, because it's a fast, is only about 40 minutes. And so all total, it's about 70 minutes, 75 minutes to be able to get your answer then. And then depending on how much you want to scale up, that would increase depending on the number of plates that you wanted to process.
Thank you, Laura. And I think we're going to wrap with this final question here. Are the kits also useful in plant and bacterial RNA analysis for genetic modification?
That is a great question and something that I don't really have an answer for. We have very briefly looked at bacterial RNA just with E. coli. We haven't looked at any other RNAs. The challenges with bacteria are the cell wall, and I'm not sure how well the lysis buffer would be able to break some of those cell walls. And again, with plants as well, those also have cell walls. And so it would be, that would be my concern, is that it wasn't optimized to work with those. And so it may not be strong enough to really break through some of the more challenging cell walls for depending on the bacteria and the plant type that you're looking at. The other challenge with plants is that they often have inhibitors inherent into the samples, like humic acids or different things like that, that can really inhibit your RT-PCR. And so in general, those you need to have some kind of cleanup method to get rid of those so that you don't inhibit your RT-PCR. So I wouldn't anticipate it working with plant samples. I think we would probably have to design something specific for that. But potentially for bacteria, we just haven't really tested it very much. And as I mentioned, we've only tested with E. coli, and that was only just one brief experiment. So I don't have a ton of information to be able to share there, but it could potentially work with bacteria.
Very good. Thank you, Laura, for your time today and for sharing your expertise in streamlining RNA analysis. Do you have any final comments or wisdom you'd like to share with our audience before we close?
I'd just like to say thank you again for joining me. It's been a pleasure to share some of this information and data with you. If you do have more questions, we do have an easy URL that you can go to. It's thermofisher.com/cells-to-Ct. And this will give you a lot more information about other kits that I didn't mention here. And so that can be a good place to start and do some more investigation. You can also reach out to your local sales rep or reach out to technical support for more questions if you have those, and we'd be able to help find the right kit for you. But again, thank you for your time today.
And thank you, Laura. And I do want to point out to our audience, we have that easy URL right there on the bottom right of your screen. So feel free to copy and paste that into your browser. And I do want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I want to also thank our audience for joining us and for their interesting questions. Quite a few questions we were unable to get to today, but don't fret, those will be answered via the email address you provided at the time of registration. And additionally, you can submit questions during the on-demand period as well. They'll be addressed via that email address. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Laura, again, thank you so much. It's always a pleasure working with our friends here at Thermo Fisher Scientific. And to all of you out there, thank you for joining us. Have a beautiful day.